All right, these notes are on RNA. So RNA stands for ribonucleic acid. So unlike DNA, it doesn't have that deoxy, it just has ribonucleic acid. First of all, RNA is found in the nucleolus. So it's not actually in the nucleus itself, it's in the nucleolus. So remember, here's your nucleus and then here's your nucleolus. So this is where RNA is found. That's one thing that a lot of people don't remember. The other thing about RNA is that it's single-stranded. So unlike DNA, it's not going to have that double helix look. It's just going to be one strand, like half of a ladder. Its role is to take messages from the nucleus or DNA to the ribosomes to make protein. RNA has 4 bases, just like DNA, but they are different, or one of them is different. So the 4 bases for RNA are adenine, which is the same as DNA, guanine, which is the same as DNA, cytosine, which is the same as DNA. Here's the new one, uracil. So uracil is only found in RNA. So the base pairing rules for RNA to RNA are A and U go together, and then G and C go together. So G and C are going to go together all the time. When you're converting DNA to RNA, the base pairing rules, A goes to U, T goes to A, and then G and C go together. So remember that A and T go together, and there are no T's in RNA. That's the biggest thing. So there are no T's in RNA. There are 3 types of RNA. The first one is mRNA, and that's messenger. And that's going to take the message from nucleus to ribosomes. Okay, you have tRNA, which is called transfer RNA. Transfer RNA brings the amino acid to correct RNA sequence. And then the third one is rRNA, which is ribosomal RNA. rRNA is helps translate the info from the messenger RNA into a protein. We're going to talk about the 3 different types of RNA later when we get to protein synthesis, but I just wanted to introduce those so you knew about the 3 different types of RNA. And let me go ahead and draw what RNA looks like real quick, because it does look different than the DNA. So here's our strand. So 1, 2, 3, 4, 5, 6, 7. And I'm just going to do some random bases. So A, U, G, C, AUA. Remember, there are no T's. So you still have your nitrogenous bases. And you're still going to have that alternating sugar and phosphate group, except now the sugar is ribose instead of deoxyribose because it's RNA. So here's our sugar, so ribose, and here's our phosphate. So it's still on the sides alternating a sugar and phosphate group, except the sugar's different. And then you still have your nitrogenous bases for the rungs, but there aren't any hydrogen bonds because they're not connecting anything. They're just there. So that is the structure of RNA.